Good afternoon, everyone, and thank you for attending today's webinar. My name is Joel Hewitt, and I'm a subject matter expert here at the Homeland Defense and Security Information Analysis Center, or HDIAC. We are pleased to present today's webinar on investigating ultrastructural mechanisms of TBI using X-ray diffraction. Our presenters today are Dr. Joseph Orgel from the Illinois Institute of Technology, Ashley Eidsmoor and Dr. Jason McDonald, both from the U.S. Army Combat Capabilities Development Command's Army Research Laboratory. We will introduce them further in just a few minutes. As we begin, note that the webinar chat function is enabled. Please feel free to type in any questions as we go along. Also, please note that we are recording the webinar and the video webcast will be available for download from our website tomorrow. A copy of the slides will also be available for download at the end of the presentation. So HDIAC is a Department of Defense sponsored entity, one of 3 information analysis centers. Organizationally, we fall under the Defense Technical Information Center, or DTIC, and the Assistant Secretary of Defense for Research and Engineering. Our mission is to be the go-to R&D, S&T, and RDT&E leader within the Homeland Defense and Security community. We achieve this by providing timely and relevant information, superior technical solutions, and high-quality products to the DoD, and HDS, COIs, and COPs. In doing so, we are able to help solve the most challenging technical problems facing the government. We pursue this mission across 8 focus areas: alternative energy, biometrics, CBRN defense, critical infrastructure protection, cultural studies, homeland defense and security, medical, and weapons of mass destruction. Our external subject matter expert network is a critical tool in achieving this mission. And if you have expertise in one of our focus areas, we encourage you to apply. Our SMEs help us provide the military and government with the most up-to-date and cutting-edge information and innovation. So now to introduce our speakers. Dr. Joseph Orgel is a tenured professor at the Illinois Institute of Technology, with past and present appointments in biology, physics, biomedical engineering, and applied health sciences. Orgel leads investigations of brain pathological diseases, such as Alzheimer's and TBI, in collaboration with ARL. He also investigates connective tissue conditions, including heart disease and arthritis, at the National Institutes of Health Biotechnology Research Resource, BioCat, as Associate Director. Ashley Eidsmoor is an electrical engineer and brain injury researcher at the Army Research Laboratory and has over 10 years of experience working with the Department of Defense on brain injury-centric technologies. She holds both a bachelor's and master's degree in electrical engineering from Purdue University, with a focus in biomedical imaging and sensing. Her thesis work focused on the development of a theory for the establishment and maintenance of neural communication within plastic brain regions based on finite state machine ad hoc network routing and stability convergence. And Dr. Jason McDonald joined the Army Research Laboratory in 2012, has been involved with computational work, primarily finite element analysis, and problems ranging from ballistic response of ceramics to cavitation in soft materials. He received a BS in physics from Oregon State University in 1991, and a PhD in physics from Iowa State University in 1997. And with that, to our presenters, we turn it over to you.
Thank you, Joel. So hello, my name is Ashley Eidsmoor, and I am here to talk about the brain, as well as the work my colleagues and I are doing to better understand the causational mechanisms from which brain injuries occur and impose lasting physiological effects. So traumatic brain injury happens when a bump, blow, jolt, or other head injury causes damage to the brain. Every year, millions of people in the U.S. suffer brain injuries, and more than half are bad enough that people must go to the hospital. The worst injuries can lead to permanent brain damage or death. These injuries begin when an initial insult, such as a car crash, sports collision, or gunshot wound, and the initial insult results in physical brain trauma resulting from displacement of the physical structures of the brain. Physical trauma to the brain then initiates these secondary processes occurring in the minutes, hours, and days following the primary injury. Secondary injuries can include breakdown of the blood-brain barrier, leading to further escalation of inflammatory pathways and even cellular death. Symptoms of TBI can be mild, moderate, or severe depending on the extent of damage to the brain. And typically the symptoms of TBI fall under several categories: cognitive, emotional, and physical. Cognitive symptoms include compromise, difficulty with attention, memory impairment, and mental fog. Emotional include psychiatric disorders such as depression, anxiety, and post-traumatic stress disorder, whereas physical symptoms might include chronic pain, migraines, and sleep disturbance. From the DoD perspective, it is important to note that while brain injuries were the highest cause of immediate fatalities during the recent conflicts in Iraq and Afghanistan, the majority of sustained TBIs in the military are mild. With respect to the warfighter, outcomes following head injuries can be grouped and examined in relation to the injurious risks they pose. Outcomes that elevate risk of immediate death or immediate lethality, outcomes that elevate the risk of delayed death, secondary lethality, incapacitation, and long-term outcomes that include behavioral and memory problems. As an example, a head trauma that results in cessation of breathing, or apnea, as it's called, may be immediately lethal in a combat scenario, whereas receiving the necessary and potentially complicated treatment in time is unlikely in these scenarios. But a head trauma that impairs but does not cease respiratory activity in comparison may not cause death but can increase the difficulty in treating a polytrauma scenario, such as if you were not able to form clots in your blood as well. The study we'll be discussing today concerns the final of these groups, which is long-term outcomes. Specifically, we're focusing on better understanding the causational mechanisms of TBI-induced myelinopathy. This outcome is seen to affect the performance and cognitive abilities of suffering individuals and can lead to decreased mission performance of the warfighter, as well as an associated decrease in overall quality of life. So what is myelinopathy, and how does it lead to these deleterious effects? As a quick overview, neurons in the brain, which are the cells that are within the brain, are wrapped in layers of fatty material called myelin, similar to how a wire can be wrapped in an insulating material to facilitate rapid signal transduction and to prevent signal degradation. Myelinopathy is the degradation and proper structuring or loosening of these myelin sheets, leading to worsening intraneural communication and potential changes in function and behavior for the sufferer. The presence of myelin degradation has been shown in individuals suffering from a host of psychological disorders, including depression and post-traumatic stress disorder. For the warfighter, we focus on the fact that this degradation can be brought on by direct head traumas. In a study on collegiate hockey players, individuals were found to have suffered widespread myelin damage and performed much lower on cognition tests, and were more likely to sustain chronic concussive outcomes in response to injury compared to their peers. While myelin damage in the form of loosening could be detected via magnetic resonance imaging, or MRI, 2 weeks post-injury, the progression of myelin damage from the initial impact to this detectable state is not well characterized. Localized structural changes to myelin that occurred during this time period may be an indicator of concussive outcomes, which would make it a useful criterion for the onset of TBI. Furthermore, deeper understanding of the instigation of myelinopathy in a traumatic brain injury situation may allow us to help treat or even halt the progression before a decreased cognitive effect is observed. I will now hand it off to my colleague, Dr. Jason McDonald, to introduce the team and partnerships which were formed in an effort to investigate potential initiating mechanisms of myelinopathy and traumatic brain injury. Jason?
Oh, hi. Just in the way of history of how the project started and the team came together, Ashley and I are members of a branch here at CCDC ARL whose primary mission is soldier protection, and a big part of that is head protection. So the design of better head protection technologies would be greatly aided if we had a better understanding of how these mild brain injuries initiate and develop following mechanical insults such as blunt impact or blast loading through processes like myelinopathy that Ashley was describing. So a few years back we were discussing what experimental methods might be used to study these types of processes, which is obviously a very challenging thing because the techniques would need to be capable of detecting structural changes to subcellular entities and their molecular constituents. So you're talking about length scales, ranging from nanometer up to micron size. So there aren't many techniques around that are capable of that. Now, one we were familiar with was X-ray diffraction. Here we were more familiar with using it for traditional materials like metals and ceramics, even fiber composites, but we didn't really have any background in applying it to studying biological tissues. So we began to look into it, and to make a long story short, we were eventually led to Joseph and his team because of their past and present work they were doing and the reputation they had for their unique capabilities. So Joseph has 25 years of experience in structural biology and fiber diffraction. Rama, at the time, was his PhD student and did a substantial portion of the work as part of his thesis. He's since graduated. And Dorothy has vast background in TBI research and provided the samples used in the study. So on the next slide, basically Joseph is, in addition to being a professor at the Illinois Institute of Technology, is also the associate director at a facility called BioCat, which is actually the leading research facility in North America for biological fiber diffraction. It's one of the sectors located at the Advanced Photon Source at Argonne National Laboratory outside of Chicago. So the Advanced Photon Source is a synchrotron, or a type of electron accelerator, which produces intense X-rays that can be used to study all kinds of materials. So, like at ARL, we were already a big user of another sector called the dynamic compression sector that we use to study shock propagation and damage in more conventional materials, but it wasn't set up to do the biological problem. Now at BioCat, they have specially designed detectors they made themselves to study structure and dynamics of biological materials over a large range of length scales, which is exactly what we needed for this study. In addition, they've also developed lots of novel imaging and scanning technologies, which they can bring to bear. So at this point, I would turn it over to Joseph so he can better describe what they actually do and how powerful it is.
Thank you. So as Jason was alluded to, fiber diffraction diffraction that's collected from materials that are both biological and not necessarily crystalline have a tendency to give at least some kind of insight, some kind of information into into how the material is put together, even if it is, as I already said, non-crystalline, such as you may well find in the organization of biological tissues. Now, it may seem oxymoronic if you know anything about X-ray diffraction or diffraction in general, for something that isn't crystalline to give diffraction. But this is because the order that is necessary, the repeating order that is necessary for diffraction to occur, exists at the levels, various levels of biological molecular structure. So, for instance, in this diagram, this schematic is giving a representation of the helices in various parts of the tissues, for example, and the repeating elements of the helices is that which gives rise to the non-crystalline diffraction, and that's something that's at least passingly familiar to people who have some kind of exposure to fiber diffraction. However, so if you look at that slide currently, the red image is giving a bit of an impression of what some of the complications are in terms of interpreting this kind of data, and that is I've just talked about non-crystalline, but there also tends to be crystalline domains as well, and that diffraction will lie on top of each other. So that complicated schematic on the right is giving you an indication of different components, different types of diffraction that are arising due to different orderings, if you like, within the tissue. And on top of all of that, those crystalline domains, those crystallites, will be orientated all the way around the clock in all sorts of orientations within what is usually a fiber or a fibrous organization. Fortunately, it is possible to make use of key elements in that diffraction pattern that we could call characterizing diffraction to pull out key insight into particularly significant bits of that organization. So, for instance, for highly organized fibrous crystallites, such as type 1 collagen, as commonly found in tendons, ligaments, bones, skin, actually the basis of most mammalian organs, that left picture is showing a fiber diffraction diagram. It's a highly ordered tissue, and it's a lot more ordered than some of the diffraction. It's a lot more ordered than the diffraction that we obtain from the neurons in the rest of the study, but it gives a little bit of insight as to how far this can go under the right circumstances. So the diffraction on the left is arising from the fiber, or the collection of fibers of collagen in element A, which contains all of the structure that you see in B, C, and D. All of those structural levels are combining to give the diffraction that you see on the left. However, even though it's very complicated, that middle section of what we call meridionals through the vertical central section of the diffraction pattern is not overlapping with anything else, and is actually quite straightforward to extract, and gives a great deal of insight into what's occurring through all levels of the structure. And that's kind of the principle that we take for the diffraction that's arising from the myelin of the neurons that we used in our study. Now, one of the great things that Jason mentioned about X-ray diffraction, as opposed to single crystal crystallography or a variety of other very important characterizing techniques that we make use of in life sciences and structural biology in general. One of the really good things about X-ray diffraction in general is it has the ability to access a length scale that's very broad all at once, from the angstrom all the way through to the micron level, potentially even on the same X-ray diffraction picture. And then you can bootstrap, if you like, a little bit further to get even higher or lower resolution domains through the use of combining the techniques multimodally, or through something that we did later on in the study, which we called X-ray diffraction scanning. Now, in the case of the neurological aspects of tissues that we study, such as obviously single isolated neurons, or for sections through the brain, there are multiple components of those tissues and those cells that are contributing to the X-ray diffraction pattern. So you see at the top of this image we have a myelinated nerve, and you can see that it's myelinated in the way that successive layers of myelin are laid down by myelinating cells. And it's that successive series of layers, that packing, that gives rise to the X-ray diffraction. So you see on the right, you have a cross-section through the myelin layers, and you can see the dark and the light banding pattern through there. And then that insert, which says 40 angstroms and 80 angstroms, is actually some 60-year-old diffraction. But that 40 and 80 angstroms, those samplings are coming from the diffraction because of the regular spacing between the myelin layers that you can see in the electron micrograph. So in the lower part of the slide here, we're showing a little bit of insight into using the synchrotron radiation source, a certain amount of focusing of the X-rays that are coming from the synchrotron through our optics, a certain amount of collimation, and a little bit of focusing. And then a lot of detail goes into the sample or alignment with the beam and so on, and then the detector gives us an X-ray diffraction, which is somewhat represented by the lower bottom right of the more recent diffraction from, I believe that's rat optic nerve. But at the end of the day, it's as simple as basically putting the nerve in front of the X-ray beam and collecting the X-ray diffraction data. And that's one of the very good things about the technique, is it enables us to do minimal sample preparation. In fact, most of the sample handling for these projects are concerned with keeping the tissue as close to the state as it was in when it was in the animal as it is now actually in front of the X-ray beam. So the sample preparation and the potential for sample preparation artifacts are actually, I would argue, very low. And this is part of what this image is here to demonstrate. What happens when you do attempt to artificially maintain the tissue in the state at which it was in before it was extracted. So on the left of this composite X-ray diffraction picture, is fresh is a subjective term, but what we mean here is it's not fixed, it's maintained hydrated, it's maintained in high humidity in the air environment. And on the right, this is a glutaldehyde fixed representation. And the 80 angstrom reflection that's shown there, it's the second order of the myelin diffraction, is altered to the 63.5 angstroms or so because it's become dehydrated in the fixation application. And the fixing, and even this is a very harsh method of fixing, the fixing may be very useful for techniques that don't penetrate to this level of resolution. And the thing is, as we find out in this study, this change that occurs with the sample treatment here is right in the region of the changes that occur to myelin packing on that initial impact that leads to that first structural change. So in this slide we're showing in the top left a much more recent X-ray diffraction picture. The optic nerve is orientated as the arrows indicate there in the axon direction, and then the diffraction from the image that arises from the myelin packing is perpendicular to that, and it's labeled. And you can see the second of the fourth orders in that particular picture. So what's happening here is the X-ray beam is generating the diffraction through that successive packing of myelin layers, as is indicated on the right-hand side there. And changes in the packing that could occur due to water influx or water efflux, or possibly changes in those transmembranous protein organizations and changes in those protein and protein lipid domain organizations. Those are detected by the X-ray diffraction, and when that's coupled with a controlled study of maintaining a careful read on how much force or how long the force is applied and so on, coupled with the X-ray diffraction, gives us a potentially very powerful mechanism for studying when change occurs and under what circumstances it occurs, and then linking that potentially to behavioral and cognitive changes that occur in people who show similar changes in their myelin layers. So this image is showing 160 and 80 angstrom spacing. The 160 is the principal packing domain between the dense myelin layers. The 80 is between the interdomains, and this means that the even orders, the second, fourth, and so on, from myelin diffraction are very strong, but the first and third, the odd orders, are very weak, in case that was a question for you.
Thank you, Joseph. So in order to initially observe the effects of mechanical loading on neuromyelin, we utilized a rodent optic nerve model, as Joseph was describing. And this is because the optic nerve is densely myelinated, and it's simple to promptly extract post-mortem. Additionally, there have been reports of perturbations to optic nerves in animal models of traumatic brain injury, making it another ideal region to examine. So what we did is these rat optic nerves were dissected and they're incrementally loaded using an impact device to examine when mechanical loading influenced the periodicity and the long-range ordering of the myelin. So we had 3 impacted groups and a control group, each comprising a total of 5 optic nerves, which were scanned using X-ray diffraction at 100 scan points each. Now Jason will describe the device which was constructed to deliver the quantifiable loads to these extracted optic nerves. And then following that, Joseph will further describe some of the interesting results that we were able to see.
Okay, so the device that was custom designed to deliver the quasi-static loads that were described in the last slide is depicted on this slide here. So a crushing force was delivered by a cylindrical head that's about 3 millimeters in diameter and driven along a linear slide by a stepper motor, which can be programmed to deliver different force magnitudes for a duration of about 1 second. So this particular slide is depicting a case where the sample is an extracted optic nerve, but the same device was used also to load brain slices. Now, since the diameter of the impactor head is so much larger than the sample size, we could assume uniform loading over the cross-section of the sample. And the sample was loaded on a glass slide and then transferred and sandwiched between 2 mica sheets for the actual diffraction measurements. So the typical plots of the forces it was producing are shown in this bottom plot. for 3 different levels of force. So you can see it's pretty uniform over that 1 second time. So now I'll turn it back over to Joseph, and he can describe the results that are obtained from these loaded samples in the diffraction measure.
Thank you. Yes. So in this slide, there's an awful lot going on in here, and I'll walk you through it somewhat. The green and the black chart in the top left is actually data that's collected across several hundred individual experiments, and they're representing 2 different phenomena that are observed in the X-ray diffraction of the myelinated nerve. And in the top, this from the green line is showing the progression downwards in terms of packing, so a shrinkage of the distance between the myelin layers that occurs at what seems to be a threshold loading force. So you can see at 10 grams load, there is no change, or essentially no change from the control group. And then at 15 grams force, there seems to be a little bit of a step, but you see that the range of measurements increases greatly with the range bar indicating there. And then absolutely without question at 20 gram force, there is definitely a change downwards. And then with the black line, there's a different characterizing diffraction that is distinguished from the myelin packing layer that bears a lot of resemblance to neurofilament diffraction. It seems to be coming from along the direction of the cytoskeleton, which is along the axon length of the neuron, and that doesn't change in the same way. Obviously, there's a downward progression because we are crushing these samples, so we would expect that entirely. But the relationship is not the same as that we see for the myelin. Then on the right of the image, we have some representative raw diffraction, and you can see the progression from control 10, 15, and 20 grams loading. And then perhaps a little bit more directly and immediately useful to you in this image is the line graph at the bottom. That's showing the second and the fourth orders of myelin diffraction, that's X and Y respectively. And you can see the peaks are shifting downwards, both those diffraction events indicating a reduction in the packing levels, but also you can see that the intensity, so that's the height in arbitrary units on the y-axis, the intensity is reducing as well. And that's indicative of the composite crystallites, the fibers as it were, or the packing in this instance, changing direction a little bit and also becoming much less coherent.
So as Joseph mentioned, we were able to observe that a notable and repeatable change to myelin structure was occurring. And in general, when we convert to PSI, this was seen to occur between 11 and almost 15 PSI, which is interesting because this range is notable given it closely resembles a pressure range of 11 to 15 PSI, which has been reported in the literature to induce secondary TBI pathologies. Additionally, we believe that the observed decrease in second-order despacing of myelin was the result of reduced water content between the membrane layers. And you can see in this image here that we're providing, this is between where you have this extracellular space of water, which is where you're seeing the approximate 80 angstrom, and this is moving to about 76 angstrom, which is a notable decrease that we had been observing. So we believe that the observed decrease in these second-order despacing, given the result of reduced water content between the membrane layers, is possible that scenarios that could be inducing this include the outflow of water from between the layers of myelin directly from the mechanical load, or possibly even compromise to embedded structural proteins, which could lead to a slight collapse of the myelin packing and subsequent water outflow. We hypothesize that these forms of disturbance would potentially have further impact on extracellular protein structures necessary for cytoskeletal and myelin maintenance, such as axoglial cams, including neurexins, neuroligands, and even nectin-like molecules, located in this narrow gap between the axonal membrane and the surrounding myelin sheath. And you can see them a little bit in the orange as a representation. So such disturbances would further compromise long-term capability for restructure and thus foster an increased risk for chronic injury outcome, including myelinopathy. but more work would be needed to better identify the timing of such events. While this developed technique allows us to view changes to myelin in small sections of optic nerve, ideally we would like to be able to extend such application to examine changes to myelin in small sections of optic nerve. In order to scan larger sections of brain to better determine how myelin injuries actually localize. So to scale up from these small areas to full brain. But we need some first steps in order to do this. So due to a generous donation of rodent brains from our partner Dorothy, we were able to develop and demonstrate functionality of a new methodology to scan larger sections of brain and observe myelin structure. So whereas Dorothy's experimental model was not able to allow us to observe the initial state structure of myelin following injury, given they were more focused on pharmaceutical applications, so just looking at the initial controls and then 3 and 30 days, we were able to show preliminary findings on how myelin structure changes at later time points, which were the 3 and 30 days, post-closed cortical impact injuries, and see how this compared to results noted via other imaging methods. So as a little bit of background, closed cortical impact injury is a mechanical model of TBI that was developed nearly 30 years ago to study TBI mechanisms and also develop therapies. So closed cortical impact injury is delivered using a device that rapidly accelerates a rod to impact a surgically exposed cortical surface, following the removal of a small section of skull. This device you can see in the middle of the slide here. And in our case, we are impacting the forelimb area of the caudal motor cortex, as you can see on the slide, where the impact region is highlighted in blue. And that's shown in a top-down view of a rodent brain. So specifically, this exam was used to study rodent motor response to these injuries and to examine the associated myelin changes. So motor response impairment we were able to determine by training rodents to reach for food pellets. And in this right section, you can see the rat, which is reaching out for a pellet of food. And if the rodent becomes motor impaired after injury, they will no longer be able to quickly and successfully grasp the pellet. So I'll hand it back over to Joseph.
Thank you. So building on the initial experiments with isolated optic nerves, and I should explain briefly that this was a very intentional progression of experiments wanting to, our desire to characterize the structure to insult response, if you like, the dose and the response of each element that would be relevant to that which causes mild moderate TBI. We isolated tissue elements, isolated tissues, then isolated components of complex composite tissues as we gradually build up towards whole organism studies. So having done the isolated optic nerve work, we were well set up in terms of the X-ray diffraction methodology to progress to something somewhat more complicated. So the top of the image here is showing essentially that actually the X-ray diffraction methodology didn't change at all. The detector system, the X-ray admittance and the focusing and the optics and so on, that was all the same. The bit that changed next for scanning brain slices was how we handled the sample. It would be at the same position, kept in the hydrated state as much as is possible under the circumstances, and then maintaining its sample position through custom-built apparatus that we made at the Illinois Institute of Technology. And here is some of that initial data from the brain scanning. You see in row A, for each of these columns, you have these composite images that look somewhat like an X-ray radiograph or a cross-section micrograph of rodent midbrain, because it is. This is a scanning image that is made up from, it's a composite image made up from individual X-ray diffraction patterns that are collected at each of those points where you see something that looks like a pixel in A. If you go to B, you'll see that each of those square pixels are actually the aforementioned X-ray diffraction pictures that are somewhat reminiscent of some of the images that we showed you earlier. And the arrows are marking, and the line is running across the myelin diffraction. So you see in column C, we give you in the line graphs there the key peaks of the second order. I should mention part of the reason why we did the fixing control experimentation is to build towards the more complex experiments later on. and characterize what kind of fixation method does what. And I'm very pleased to say that we were able to find that perfusion fixing of the organisms, as is used here, produced very small changes, the smallest changes we found of any fixing method, in of the region of 5% or less, which was slightly smaller than the changes that we were observing. But most importantly, those changes were ubiquitous across the brain, whereas particular changes that we see in these images are located in particular areas. Could you go back to the previous slide? Thank you. So you see in the first column we have the control organism showing that 76 angstrom spacing. On day 3, so this is 3 days after that impact, we're at the 82, and then on day 30 we show these are global readings across the brain. We show an average return to about 80 angstroms. But if we go to the next slide, you'll see that the changes are actually concentrated in particular areas, and that's for a good reason, because the insult was delivered to one hemisphere of the forelimb control region of the motor cortex. So in this image, you might, with the eye of faith, be able to see something that isn't there, because this is a control animal. The control animal here, we're able to take that scan, the X-ray diffraction data, and then use meta-analysis to determine 2 different things. One, orientation of the myelin, and the orientation of the myelin therefore tells us the orientation of the axon, so we can pick up on those changes, and then also intensity changes. How much, so intensity changes, but then in this particular image, the D-space changes that we've translated into a grayscale heat map here. So increases in D-spacing, as you may remember, relates to an increase in the packing, so those myelin layers are further separated, and a shrink in the D-spacing will mean that the distance between those packing layers is decreased. Next slide, thank you. So this is one of our take-home observations from the use of these donated brain slices. In the control, we can see, as already mentioned, there's a uniform 5% disturbance in structural spacing across the brain, and then 3 days post-injury, we see that there is a brain-wide, or at least in the section, there's an increase in the D-spacing, so the myelin packing is increased throughout the entire brain, and then very intriguingly, at 30 days post-injury, that swelling has returned down to the control levels, except the average is increased because of one particular area in the brain that still has a heightened or increased packing distance, which could be interpreted as loosened myelin, but is certainly indicating that the myelin layers are now spaced apart somewhat more. And that area was actually both hemispheres of the forelimb control area of the cortex, inclusive of the area that was not actually impacted. And the reasons for this occurring is going to be very interesting to try and figure out. It could be a transmission of force along connections, tissue connections between them, and it could be due to some kind of cellular cascade event. So we're not going to be able to address that question particularly well with the data that we have alone. We definitely need to get into what's happening immediately after impact to be able to have some kind of insight as to whether or not this loosened myelination is due to the impact alone, or if it's due to the cellular cascade, or if it's due to 30 days of compensatory behavior on the part of the organism, and what we're seeing is neuroplasticity in the other hemisphere taking control for the damaged hemisphere. And this is going to be very interesting and very potentially useful to understanding how to deal with the consequences of mild and moderate TBI for humans in terms of interventions and therapies. So we're in the process of building upon this initial work. Another very useful aspect of the X-ray diffraction scanning methodology is that with the experience we have now across multiple decades of using this system, we're able to characterize what kind of biological material is giving what kind of diffraction more often than not. So diffraction coming from collagen, from myelin, from the cytoskeleton, even diffraction that may be coming from amyloid plaques or more organized organization of amyloid and amyloidoses. These are things that we're somewhat experienced with, and we're able to map in the X-ray diffraction scan, but also we're able to, as we've shown with some of the earlier images, able to go into each of those areas of the scan, anywhere that we've hit with X-rays, and pull out potentially angstrom level information as to what might be occurring in that area, and under ideal circumstances actually do a true structural solution as well. We can use principally the same equipment to collect X-ray fluorescence at the same time, which is extremely useful for picking up on what kind of metal, or as we annotate in this particular image, calcium. We can go down to about calcium size in terms of X-ray fluorescence. And that gives us further color and flavor to determine what may be happening, particularly in cellular cascades and altered genomic response to impacts. And then something that we've been doing over the last couple of years as well is combining that with other techniques, such as microscopy techniques, which we're in the process of doing for our collaboration also with the Army in characterizing heart valve behaviors, tissue behaviors, and tissue responses, particularly in composite regions where one tissue is in the process of becoming another tissue, which is very, very relevant because the mechanical behaviors and capabilities of those tissues can change on orders of magnitude in very small spatial regions. So at the moment, we're continuing to work towards ex vivo human tissue data collection. We've been doing this using essentially the same X-ray methodology for human hearts, and if NIH or if the reviewers of NIH smile upon us in the very near future, we will move on to the ex vivo human optic nerve work to translate that which we've done for the rodents to be more directly relevant to Homo sapiens, and then build the in vivo animal models, particularly that time equals 0, the right after impact, to get some insight as to what's occurring due to cellular cascade, what's happening due to the trauma itself, and all the things in between that. But then also to look at getting a finer grade of force application, of dose, if you like, to try and elucidate what else may be happening in terms of the packing changes, and then also looking at acceleration ranges. Now, the motivation for this is to generate a quantified injury outcome assessments for patients, and to use these using computational methods and accumulated data, ultimately looking at ideal options for intervention and for therapy.
Thanks again, Joseph. So from the Army end, the capabilities that we've developed with Joseph and his team have allowed us to really explore the initiating damage mechanisms in the brain in a way that may become truly beneficial to the prediction and prevention of traumatic brain injury. So future investigations would allow us to determine whether damage to proteins within the layers of myelin seem to change in response to the initial loading, whether or not they instigate myelinopathy, and that would help with getting those day 0 samples. But such findings would better direct pharmaceutical interventions in order to block these progressions and developments, such that we could angle them in order to go towards the proteins that are becoming initially compromised during these events. Furthermore, if we tie together the mechanical forces which are able to promote these initial and instigating changes, then we can develop better predictive models of injury, initialization, and progression that can be utilized for consideration in the development of new armoring solutions and even patient treatment. And you can see in the slides on the right-hand side, these are some models that the Army develops in order to look at how different changes in myelin are occurring over time and how that may tie to behavioral outcomes. And what we use is the MRI images that are showing the degradation, but it's really important to tie these initial mechanical disturbances and understand how that later degradation is occurring, such as what you'd see at the 3 and 30-day times what is actually instigating that at the initial time point. And that will really help steer how we can put the physics into these models in order to describe and predict what's happening in these later time points.
All right. Well, with that, thank you so very much. We have one question from Kyle Geisler, and anyone can answer it. How do you prevent the sample from drying out on the XRD mount during data collection? Does desiccation cause similar structural changes in the absence of an applied force?
Thank you. Yes, I'll be happy to take that. I think as we covered this earlier on, that this is prompted from some good insight as to exactly what is occurring. So the answer to this multiple composite question is yes. Desiccation, drying definitely does lead to similar structural changes, which is why we took such great pains to control the experiments in the way that we did, and actually obtain data from dried samples as well. The good thing about the X-ray diffraction is that it's apparent in real time. Looking at the raw data alone, you can tell when a sample is becoming dehydrated. And so once we learnt the circumstances and the timing under which a sample may undergo that, then we learnt the parameters that the experiments needed to be conducted in and made sure that they were done so that that didn't occur. But in general, how do you prevent it? It's maintaining the sample in that hydrated environment. Primarily, actually, it's making sure there's humidity and making sure that you're prompt.
All right, thank you. Well, we are rapidly approaching 3 o'clock, so we will call it there. Joseph, Jason, Ashley, thank you so very much.
Thank you.